Hey guys, it's Medicosis Perfectionalis, where medicine makes perfect sense, and today we'll continue our biology playlist. In previous videos, we talked about the innate immunity. Today, it's time to turn our attention to the adaptive or acquired immunity. It's time to talk about these lymphocytes. B lymphocytes for humoral immunity. T lymphocyte is for cell-mediated immunity. This is my biology playlist. Please watch these videos in order. This is a very basic discussion. For more details, check out my physiology playlist as well as my immunology playlist. What's immunity? It's the ability of your body to resist microorganisms or toxins. As you know, we have two main types of immunity. We have innate immunity and adaptive or acquired immunity. Innate is something that you are born with, not specific, less potent, and has no memory. Adaptive is something that you acquire, that you train as you mature. More specific, more potent, develops after birth, and you have memory. Then the adaptive immunity is subdivided into naturally acquired and artificially acquired. Naturally acquired, I got it from nature. All right, I could be passive or active. What do you mean by passive? I got pre-formed, pre-made antibodies from mommy, whether through the placenta or through breast milk. I did not do anything active. But here I'm active. I was actually infected and then actively developed antibodies. These were not pre-made, pre-formed. These are newly made, newly formed antibodies against the organism. So I was active here. All right, this is nature. And then we have artificial. This is the doctor's job. This happens to you at the hospital, not in nature. Example, you could be passive or active. I was passive. The doctor gave me antibodies that were pre-made by another patient and then added those antibodies to my body. They could be made by another patient, in a horse, or in a lab. Or it could be active. The doctor gave me a vaccine, which is a weakened or a killed organism. And then I fought that weak organism and developed immunity. I was being active. Innate immunity was discussed before. It is not specific. It is quick. It is less potent. Cells of the innate immunity are many, including microphages, such as neutrophils, eosinophils, macrophages, natural killer cells, and don't forget the complement system, because it's part of the innate immunity, but the complement also acts as a bridge between the innate and the adaptive immunity. These are the cells of the innate immunity. Pause and review. Today, it's the story of the adaptive immunity. We have two types, humoral and cellular. What do you mean by humoral? Oh, right, it means fluid. Humors are fluid. The ancient Greeks had a theory of the four humors of the body: blood, phlegm, yellow bile, black bile. Humors means fluid. Why do you call this humoral immunity? Because the B lymphocytes will mature to give you plasma cells to secrete antibodies in the fluids of your bodies, especially plasma, with the exception of IgA, which left the plasma and ascended to the mucous membrane surfaces that line your mouth, your nose, anal canal, vaginal canal, etc. Versus cell-mediated immunity, where your T lymphocytes is gonna fight the viruses, the fungi, and the cancers cell to cell, face to face. That's why we call it cellular immunity. Quick review. Your blood is made of plasma and cells. The plasma is water and proteins. The proteins are albumin and globulin. Don't forget that your gamma globulins are the actual antibodies made by plasma cells, which was a B lymphocyte. What else do we have in the blood? Blood cells, such as red blood cells, platelets, and white blood cells. White blood cells include granulocytes and non-granulocytes. The granulocytes are basophils, eosinophils, neutrophils. The non-granulocytes are monocytes and lymphocytes. Blood is made of plasma and cells. Plasma is made of water and proteins. Proteins are albumin or globulin. Globulin has many subtypes, including gamma globulin. The antibodies are here. You can call them gamma globulins, you can call them immunoglobulins, or simply antibodies. This is the normal distribution of the globulin subtypes. An exam question can ask you the following. A patient became like this. Instead of normal, there is a peak. What does that mean? It means that gamma globulins increase in the blood. Oh, this patient has too many antibodies. Yeah, I wonder why. Maybe because this patient is fighting an infection right now? Oh yeah, that makes sense. Functions of white blood cells are many. Neutrophils, for example, fight bacteria. They also make pus. That's why we call them pus cells. They are the cells of acute inflammation. Lymphocytes fight viruses and fungi. They are the cells of chronic inflammation, not acute. These are the cells that make the granuloma. These are the cells of adaptive immunity. These are the cells of type 4 hypersensitivity, especially the T lymphocytes. Next is monocytes. In the blood, they are just monocytes. When they go to tissue, they are called macrophages. They are phagocytic cells. They can eat bacteria and others. They are also one of the antigen-presenting cells. Eosinophils fight parasites. Basophils release histamine. This is the allergy. This is type 1 hypersensitivity reactions such as bee stings, such as asthma, etc. As you know, the cells of the adaptive immunity are the lymphocytes. Early, those lymphocytes are naive. Late, they mature. How come? How do they mature? They recognize the antigen and they grew the French toast up, making the second response faster and stronger. The adaptive immunity has B lymphocytes and T lymphocytes. Let's start with the B lymphocyte, humoral immunity. B lymphocyte is going to mature, is going to recognize the antigen and grow up, becomes plasma cell. Plasma cell is going to secrete antibodies, aka immunoglobulins, also known as gamma globulins. Each immunoglobulin has two pairs of chains. We have two heavy chains and two light chains. The heavy chains could be M, A, G, E, or D. That's why we say IgM, IgD, IgG, IgA, etc. The light chains could be kappa or lambda. So you pick one from here and one from here. For example, this antibody could be IgM lambda. The structure of the antibody. As you know, we have two heavy chains and we have two light chains. Between the two heavy chains, there is a disulfide bond. This is the variable portion because it differs from an antibody to the next, but this is the constant domain, C for constant. Where does the antigen bind? Here, this is the antigen binding site, and therefore you will have an antigen-antibody reaction. What if the macrophage wants to come and eat the entire thing? Well, the macrophage is a cell. It's going to be here, near the constant portion. So antigens bind near the variable portion, but cells bind to the constant portion. Here's the bacteria. Take a piece. That's the antigen, which is a piece from the bacteria. Antigen is going to bind to the antibody. Where? On the variable portion. Okay. How about the constant domain? Well, the C is bound to the C, the cell. And then the macrophage is going to eat everything. It's going to eat the antigen-antibody complex. What's the function of complement? It complements the ability of antibodies to do their job. What types of antibodies do we have? IgG, IgM, IgA, IgD, IgE. Who made them? The plasma cells, which were B lymphocytes. The nature of these antibodies are gamma globulins. Let's go. IgG. I am the most abundant immunoglobulin in the circulation. All right. I'm responsible for the secondary response to the bacteria. All right. Who's responsible for the primary response then? IgM. Okay. IgG. All right. Even though I am the most abundant in number, I'm actually very small in size. I'm a monomer, just one particle. That's why I can cross the placenta and cause trouble for the baby. I also help make the bacteria tasty, a process known as opsonization. Why do you want to make the bacteria tasty? So that the phagocytic cells enjoy themselves while eating the bacteria. It makes the secondary response faster and stronger. Next, IgM. I am the primary response. I am large. I am a pentamer. So IgG is the most abundant. IgM is not that abundant. IgG is secondary response. IgM is primary response. IgG is very small, a monomer. IgM is large, it's a pentamer. That's why it cannot cross the placenta. IgM is very good at fixing complement and activating the complement system. Next, IgA decided to ascend and leave the plasma, leave your blood vessels, and go to the mucosal surfaces. In your nose, in your mouth, in your ear, in your paranasal sinuses, in your gut, in your anal canal, vaginal canal, etc. IgD, no one knows what it does. IgE, just remember ew, allergies, ew, parasites, ew, anaphylactic shocks with histamine, ew. Here's a quick note. When the B lymphocyte was naive, she was a doctor, just MD. It can only secrete two of them, M and D. That's why we say produced first here. However, once we recognize the antigen and grow, oh, that's different. This is like 5 PhDs. I can now secrete anything you want. I can give you IgG, IgM, IgE, IgD, IgE. This maturation is called class switching because I went from only producing M and D to being able to produce everything. Class switching. Switching the class of the antibodies. The antigen is going to bind here. The cell is going to bind here. The complement binds here because the complement complements the ability of the antibody to perform its function. What happens when the antigen and antibody hug each other? They can cause neutralization of the bacteria and bacterial toxin. Opsonization, make the bacteria tasty so that we can eat it better. Agglutination of antigen antibody. Activation of natural killer cell and activation of the classical pathway of the complement system, which complements the ability of antibody to perform its functions. The story of immunology. Alright, here is a bacteria. Take a piece, it becomes an antigen. Who's going to present the antigen? The waiter. Who's the waiter? Antigen presenting cell. How do you present the food? I present the food, antigen, on a tray. What's the name of the tray? MHC. Major histocompatibility complex, which was discussed in the previous video. Who's the diner? Who's the customer waiting for the food? The lymphocyte. Once the lymphocyte sees the antigen, i.e. the food, the lymphocyte has options. It can eat it, phagocytosis, remember it, memory cells, or change into plasma cells that secrete antibodies if this is a B lymphocyte. B lymphocyte, humoral immunity. Why? Because I was naive. I was just M and D. Then I matured. Then I class switched. Then I can give you plasma cells, which can give you all kinds of antibodies that you want. I will also remember this infection so that my secondary response for the same bacteria is going to be stronger and faster in the future. Versus cell-mediated immunity. T lymphocytes, cell-to-cell, face-to-face destruction of cancer cells, fungus cells, and not just viruses, but virus-infected cells. If a virus infected my own cell, I'm gonna kill the virus and the infected cell that belonged to me. I will destroy everything. I am cellular immunity. We have many types of T lymphocytes, including T helper, T cytotoxic, and T regulatory. T helper, what do? I help people. I help my sister, the T cytotoxic. I help my neighbor, the B lymphocyte. Help her do what? Become a plasma cell and secrete antibodies. And how do you help your sister? I help her destroy fungi, cancers, and virus-infected cells. How about the T regulatory cells? It was formerly known as T suppressors. Why? Because it suppresses those crazy lymphocytes that are going to destroy your own cells. Oh, that's not good. I want to destroy the foreigners. I do not want to destroy my own cells. Okay, so take those crazy lymphocytes that were going to destroy your own cells and suppress them to avoid autoimmune diseases where your immunity is destroying yourself. This will be prevented thanks to T regulatory cells. What's an antigen? It's a piece of the bacteria. Why do you call it antigen? Because it will generate an antibody against it. Oh, I get it. It's a foreign substance that stimulates your immune system, triggering an immune response. What's a hapten? It's a baby antigen. On its own, it cannot trigger an immune response. But hapten plus protein, now this can trigger an immune response. The MHC system was discussed in the previous video. Remember, the MHC is the tray on which the food is displayed. And we have two main types of MHC, 1 and 2. Who makes lymphocytes? Bone marrow. Where do they mature? If you are a B lymphocyte, you will mature in the bone marrow. But if you are a T lymphocyte, you mature in the thymus. Think of it like police officers. Where were they born? From their mother's womb. The mother's womb is the bone marrow. How do they train? They train in police academies. Oh, if you are B lymphocyte, bone marrow. If you're T lymphocyte, thymus. Thank you. Where are they situated and located? At police stations such as lymph nodes, spleen, mucosa-associated lymphatic tissue, and tonsils. The topic of lymph and lymph node will be discussed in the next video. Here is the story in a nutshell. Here is your lymph node. What's the function? It filters and cleans your blood from all the gunk, all the debris, all the bacteria, etc. These are called lymph vessels or lymphatics. This is the lymph node. Here's your lovely kidney-shaped lymph nodes. It has many structures inside. For example, it has this follicle, which contains germinal centers. What do they contain? B lymphocytes. All right, thank you so much. All of this is in the cortex. Oh, near the crust? Yeah, the cortex is the crust. All right, let's dig deeper. After the cortex, what do I get? You get paracortex. What does it contain? T lymphocytes. Okay, thank you so much. Can I dig deeper? Sure. You will go to medullary cord. Oh, in the medulla? Yeah, it's not the cortex anymore. It's the medulla. Cortex is on the outside. Medulla is deep on the inside. All right, we are medulla. We are on the inside. What do we contain? Plasma cells. Oh, which are the B lymphocytes. As they matured, exactly true. As they mature, they go this way because they are ready to leave to go to your lymphatics and then your blood to fight all kinds of infection. Let me dig deeper. What do I find? You find medullary sinuses. Who are there? Macrophages. So let's review. Cortex is on the outside. Medulla is in the core. Let's give more detail. We have cortex, paracortex, medullary cords, medullary sinuses. The cortex has B lymphocytes. The paracortex, T lymphocytes. The medullary cords, plasma cells, which are the mature B lymphocytes. And then the medullary sinuses have macrophages. Cortex, paracortex, medulla, medullary sinuses, B lymphocytes, T lymphocytes, plasma cells, macrophages. Can we talk about the cortex? Yeah, it has B lymphocytes living in this lovely follicle. When the B lymphocytes are immature and naive, this was called primary follicle. As they mature and grow and wise up and class switch, this becomes a secondary follicle, a more sophisticated follicle with a germinal center, mantle, and margin. We're done with the lymph node. Let's talk about another police stations, the spleen. What does police station contain? Police officers? Lymphocytes, you mean? Yeah. B lymphocytes and T lymphocytes? True. Do you remember my lymph node? Yeah. B lymphocytes were in the follicles. T lymphocytes were in the paracortex. In the spleen, it's very similar. B lymphocytes are in the follicles as well. All right, that's fair. T lymphocytes are in the, not paracortex, but peri-arteriolar lymphatic sheath. You will always find the T in the P. Paracortex, if we are lymph node. Peri-arteriolar lymphatic sheath, if we are in the spleen. The woke mnemonic that I invented is this. Trump Pence. The T is always in the P. We're done with B lymphocytes. Can we talk more about T lymphocytes? T lymphocytes are here. Cellular immunity. Who made the T lymphocytes? Bone marrow. Where do they mature? T mature in the thymus. Where are they located? Many areas, including stations, lymph nodes, spleen, tonsils, mucosa-associated lymphatic tissue, and many others. Types of T lymphocytes. We have T helper, to help sister, and to help neighbors. And then we have T cytotoxic, to kill cancer cells, fungi, and virus-infected cells. And T regulatory, to regulate and suppress those crazy cells that were going to kill your own cells. It keeps your own immunity in check. How do the T lymphocytes get active? By recognizing the antigen. Who's gonna present the antigen? The antigen-presenting cells. These could be macrophages, B lymphocytes, or dendritic cells. The T lymphocytes are many types. Some of them are CD4, some of them are CD8. That's the identification card of the T lymphocyte. For more details about this, check out my physiology playlist. How do humans communicate with each other? Well, we have the snail mail, we have email, we have phones, we have the internet, etc. But how do your immune cells communicate with each other? By releasing cytokines. Cyto means cell, kine from kinetic, motion. Put those cells in motion by sending cytokine signals. Examples of cytokines. We have interleukins, the abbreviation is IL, such as interleukin 1, interleukin 2, interleukin 6, and gazillion others. And then we have colony-stimulating factors to stimulate the colonies to grow and multiply, and we have tumor necrosis factor. Can we compare between humoral immunity and cell-mediated? Sure. Humoral is B lymphocytes, cell-mediated is T lymphocytes, and then B lymphocytes fight bacteria by secreting antibodies because they become plasma cells, as you know. How about cell-mediated? They are not for bacteria. They are for viruses, fungi, and cancer cells. They do not secrete antibodies. They go and do the killing by themselves, face-to-face, cell-to-cell. That's why we call it cell-mediated. Can you compare between B lymphocytes and T lymphocytes? Sure. B lymphocytes, humoral. T lymphocytes, cell-mediated. Who made them? The bone marrow. Where do they mature? B lymphocytes mature in the bone marrow. T lymphocytes mature in the thymus. This is called primary maturation. Then the secondary maturation happens in lymph nodes, spleen, mucosa-associated lymphatic tissue, tonsils, etc. If you're B lymphocytes, we'll always find you in the follicles. If you're a T lymphocyte, we'll always find you in the P, paracortex in the lymph node, periarterial lymphatic sheath in the spleen. Primary maturation is mostly endodermal. Secondary maturation is mesodermal. If my channel helps you, please consider buying me a coffee. Some clinical implications of the adaptive immunity. One of the most important functions of your immune system is to recognize and differentiate between yourself and the non-self. We should preserve the self. We should kill the foreigners. How do you tell your immune system not to attack your own cells? By many mechanisms. Some of them are called energy. Those crazy lymphocytes, well, delete them. And don't forget your T regulatory cell. What if I failed to prevent my lymphocytes from attacking my own cells? Well, you will get autoimmune diseases, such as type 1 diabetes, myasthenia gravis, etc. Sarah needed a new kidney. Sarah can recognize her own cells. She can recognize the self. But if she decides to receive a new kidney, being transplanted from Adam, for example, Adam is foreign to her. That's why there is a very high likelihood that Sarah's immune system is gonna destroy the new foreign kidney. That's why before we perform tissue transplant, the doctors and the surgeons gonna make sure that they are a match by matching their ABO system, RH system, and MHC. The same exact concept applies to blood transfusion. We gotta match the blood before transfusing it to make sure that the recipient and the donor match each other. Otherwise, you'll stimulate and trigger the immune system, causing red blood cell destruction and hemolysis. If you want to download these notes in PDF form, go to medicosisperfectionalis.com. To take it to the next level, check out my hematology playlist. Until next time, please watch these videos, the lymphatic system and the anatomy of the lymph node, until I see you next time in this biology playlist. If you like this video, you will enjoy my renal physiology course available to download at medicosisperfectionalis.com. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website to download my courses. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine makes perfect sense.